Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's lesson, I'm going to introduce to you the nucleus and the DNA replication. So stay tuned. The nucleus is considered as the largest and the most prominent of all cells organelles. The nucleus is generally considered the control center of the cell because it stores all of the genetic instructions for manufacturing proteins. Interestingly, some cells in the body, such as muscle cells, contain more than one nucleus, which is known as multinucleated. Other cells, such as mammalian red blood cells, do not contain nuclei at all. RBCs eject their nuclei as they mature, making space for the large number of hemoglobin molecules that carry oxygen throughout the body. Without nuclei, the lifespan of RBCs is short, so the body must produce new ones constantly. In this picture is the nucleus of the muscle cells. In the figure B, unlike cardiac muscle cells and smooth muscle cells, which have a single nucleus, skeletal muscle cells contain many nuclei and is referred to as multinucleated. These muscle cells are long and fibrous, which is often referred to as muscle fibers. During the development, many smaller cells fuse to form a mature muscle fiber. The nuclei of the fused cells are conserved in the mature cells, thus imparting a multinucleate characteristics to mature muscle cells. This is a picture of red blood cell extruding its nucleus. Mature red blood cells lack a nucleus. As they mature, erythroblasts extrude their nucleus, making room for more hemoglobin. The two panels here show an erythroblast before and after ejecting its nucleus respectively. So inside the nucleus lies a blueprint that dictates everything a cell will do and all of the products it will make. This information is stored within DNA. The nucleus sends commands to the cell via nuclear messengers that translates the information from DNA. Each cell in your body, with the exception of germ cell, contains the complete set of your DNA. When a cell divides, the DNA must be duplicated so that each new cell receives a full complement of the DNA. This time, may I discuss to you the organization of the nucleus and its DNA. Like most cellular organelles, the nucleus is surrounded by a membrane called the nuclear envelope, and this membranous covering consists of two adjacent lipid bilayers with a thin fluid space in between them. So spanning these two bilayers are nuclear pores. A nuclear pore is a tiny passageway for the passage of proteins, RNA, and solutes between the nucleus and the cytoplasm. So protein called pore complexes lining the nuclear pore regulate the passage of materials into and out of the nucleus. Inside the nuclear envelope is a gel-like nucleoplasm with solutes that include the building blocks of nucleic acids. There also can be a dark staining mass often visible under a simple light microscope called a nucleolus. The nucleolus is a region of the nucleus that is responsible for manufacturing the RNA necessary for construction of ribosomes. Once synthesized, newly made ribosomal subunits exit the cell nucleus through the nuclear pores. The genetic instructions that are used to build and maintain an organism are arranged in an orderly manner in strands of DNA. So within the nucleus are threads of chromatin composed of DNA and associated proteins. Along the chromatin threads, the DNA is wrapped around with a set of histone proteins. A nucleosome is a single wrapped DNA histone complex. Multiple nucleosomes along the entire molecule of DNA appear like beaded necklace, in which the string is the DNA, and the beads are the associated histones. When a cell is in the process of division, the chromatin condenses into chromosomes so that the DNA can be safely transported to the daughter cells. The chromosome is composed of DNA and proteins. It is the condensed form of chromatin, and it is estimated that humans have almost 22,000 genes distributed on 46 chromosomes. So in order for an organism to grow, develop, and maintain its health, cells must replicate themselves by dividing to produce two new daughter cells, and each with the full complement of DNA as found in the original cell. Billions of new cells are produced in an adult human every day. There are a few cell types in the body that do not divide, including nerve cells, skeletal muscle fibers, and the cardiac muscle cells. The division time of different cell types varies. Like for example, epithelial cells of the skin and the gastrointestinal lining, for instance, divide very frequently to replace those that are constantly being rubbed off of the surface by friction. So a DNA molecule is made of two strands that complement each other. The molecules that compose the strands fit together and bind to each other, creating a double-stranded molecule that looks much like a long twisted ladder. And each side rail of the DNA ladder is composed of alternating sugar and phosphate groups. The two sides of the ladder are not identical but are complementary, so these two backbones are bonded to each other across pairs of protruding bases, and each bonded pair forming a rung or cross member. The four DNA bases are adenine, thymine, cytosine, and guanine. Because of their shape and charge, the two bases that compose a pair always bond together. So adenine always binds with thymine and cytosine always binds with guanine. The particular sequence of bases along the DNA molecule determines the genetic code. So therefore, if the two complementary strands of DNA were pulled apart, you could infer and the order of the bases in one strand from the bases in the other complementary strand. For example, if one strand has a region with the sequence of AG, TG, CCT, then the sequence of the complementary strand would be TCA-CGGA. So what is a DNA replication? A DNA replication is the copying of DNA that occurs before cell division can take place. This is a picture of a DNA replication. So DNA replication faithfully duplicates the entire genome of the cell. During the DNA replication, a number of different enzymes work together to pull apart the two strands of each strand that can be used as a template to synthesize new complementary strands. So the two new daughter DNA molecules each contain one of the pre-existing strand and one newly synthesized strand. Thus, DNA replication is said to be semi-conservative. We have three stages of DNA replication. So the first stage is initiation. The two complementary strands are separated, much like unzipping a zipper, and then special enzymes including helicase and twist and separate the two strands of DNA. And the second stage is the elongation. In this stage, each strand becomes a template along which a new complementary strand is built. So DNA polymerase brings in a correct basis to complement the template strand, synthesizing a new strand base by base. A DNA polymerase is an enzyme that adds three nucleotides to the end of a chain of DNA, making a new double strand. This growing strand continues to build until it has fully complemented the template strand. And the last stage, the stage 3, is called the termination stage. Once the two original strands are bound to their own finished complementary strands, DNA replication is stopped and the two new identical DNA molecules are complete. Each new DNA molecule contains one strand from the original molecule and one newly synthesized strand. So the term for this mode is replication, is semi-conservative, because half of the original DNA molecule is conserved in each new DNA molecule, and this process continues until the cell's entire genome, the entire complement of an organism's DNA, is replicated. And as you might imagine, it is very important that DNA replication take place precisely so that new cells in the body contain the exact same genetic material as their parent cell. So mistakes made during DNA replication, such as the accidental addition of an inappropriate nucleotide, have the potential to render a gene dysfunctional or useless. Fortunately, there are mechanisms in which minimize such mistakes. A DNA proofreading process enlists the help of special enzymes that scan the newly synthesized molecule for mistakes and corrects them. So once the process of DNA replication is complete, the cell is ready to divide. And this ends my lecture about the DNA replication and the nucleus. Should you have any question, please write it in the comment section down below and I'll be glad to answer them all. Once again, I would like to thank you all for watching my video and for listening to my discussion. Until my next video, bye everyone!